mengenangmu, kami susuri lembaran sirahmu, pahit getih perjuanganmu, membawa cahaya kebenaran. Engkau taburkan pengorbananmu untuk umatmu yang tercinta, biar terpaksa tempuh derita, cekanya hatimu menempuh langitnya. Tak terjangkau tinggi berketimu, tidak tergambar indahnya akhlakmu, tidak terbalas segala jasamu. Sesungguhnya engkau Rasul mulia, tabahnya hatimu menempuh dugaan, sedang erti kesabaran, juang janji kemenangan, seperti namamu di dalam Al-Quran.
Assalamualaikum. Hi, we are a group of Foundation Science and Medicine students of University Sultan Zainal Abidin. We are from Group 25. Our group members are Al-Amin Hafiz, Muhammad Sarfan, Nurul Natasha, Nur Afrina, and lastly Alif Hakimi. Our presentation is about the groundbreaking work of George Beadle and Edward Tatum, which is known as Beadle and Tatum experiment. Their experiment, often referred to as the one gene, one enzyme hypothesis, it's revolutionized our understandings of genetics and the role of DNA. So let's begin with a brief introduction to George Beadle and Edward Tatum experiment.
Beadle and Tatum experiment. Beadle and Tatum experiment is a classic experiment in the field of genetics. It is laid as a groundwork for our understanding on how gene controls the characteristics of a living organism. This experiment is created by George Beadle and Edward Tatum. In the late 1940s, George Beadle and Edward Tatum conduct this experiment using bread mold, which its name is Neurospora casa. They choose this living organism because it was easy to grow in the lab and has a simple life cycle, which means it doesn't need a complex environment to be raised in. So this is a graphic that shows a simplified version of their experiment.
Conclusion of Beadle and Tatum experiment. One gene, one enzyme. From these experiments, Beadle and Tatum conclude that each gene in an organism leads to the production of a specific enzyme, a protein that facilitates a specific chemical reaction. This concept is often summarized as one gene, one enzyme. Their work paved the way for our modern understanding that genes carry the instruction for building proteins, not just enzymes. So, the principle is now often referred to as one gene, one protein, or one gene, one polypeptide. Beadle and Tatum's work was groundbreaking and earned them the Nobel Prize in Physiology or Medicine in 1958. It provides the foundation for the field of molecular genetics and helps us understand the fundamental process of life. Hypothesis of Beadle and Tatum experiment. Beadle and Tatum's one gene, one enzyme hypothesis was a significant breakthrough in the field of genetics and biochemistry. Here are a few reasons why it was so impactful. 1. Foundation for molecular genetics. This concept was a crucial stepping stone towards understanding the molecular basis of genetics. It provides the first clear link between genes, which is the units of heredity, and proteins, the workers that carry out most cellular functions. 2. Understanding genetic disease. The hypothesis helps scientists understand how mutations in specific genes could lead to specific disease. If a gene is mutated such that it can produce a functioning enzyme, it can lead to a build-up of certain substances or a lack of others, causing disease.
3. Development of genetic engineering. Understanding that each gene codes for a specific protein has been fundamental in the development of genetics engineering. Scientists can now insert, delete, or modify genes to influence the production of specific proteins. 4. Advance in drug development. This understanding has also been crucial in pharmaceuticals. Many drugs work by interacting with enzymes, so understanding which enzymes are produced by which genes can aid in drug design and development. The one gene, one enzyme hypothesis has been refined over time. We now know that a single gene can code for multiple proteins due to processes like alternative splicing, and not all genes code for enzymes. Some code for other types of proteins or for RNA molecules that perform other functions in the cell. Nevertheless, Beadle and Tatum's work laid the groundwork for these later discoveries. The minimal medium used by Beadle and Tatum in their experiments played a crucial role in testing their hypothesis and ultimately led to the groundbreaking discovery of the one gene, one enzyme concept. Here is why the minimal medium was significant. 1. Controlled environment. The minimal medium provided a controlled environment in which all necessary nutrients for the growth of the normal Neurospora crassa were present. This allowed Beadle and Tatum to observe the effects of specific genetic mutation on the molt's ability to thrive. 2. Identification of nutrient deficiencies. By growing the mold on this minimal medium, Beadle and Tatum were able to identify which nutrients its mutant strains were unable to produce on their own due to the mutations. 3. Testing the hypothesis. When a mutant strain couldn't grow on the minimal medium, Beadle and Tatum added specific nutrients to the medium one at a time. If the addition of a specific nutrient allowed the mold to grow, they inferred the gene mutated in the strain was normally responsible for producing that nutrient. This was a key step in testing the one gene, one enzyme hypothesis.
Before we end our presentation, I would like to give one hadith and one Al-Quran. The hadith is, Prophet Muhammad, peace upon him, is reported to have said, each of you is a shepherd and each of you is responsible for his flock. Hadith riwayat Bukhari and Muslim. This hadith emphasizes the concept of personal responsibility, similar to the responsibility of each enzyme for producing a specific protein or enzyme. And for the Quran, from Surah Al-Isra, ayat 17, whoever is guided is only guided for the benefit of his soul, and whoever errs only errs against it, and is no bearer of burden will bear the burden of another. This verse emphasizes the concept of personal responsibility for one action and deeds, similar to how each gene is responsible for producing a specific protein or enzyme. That's all from our discussion. Thank you.